This technique is significant because it can measure glucose uptake in specific cells in live Drosophila tissues. The FRET-based sensor provides a direct measure of changes in intracellular glucose levels.
This technique allows researchers to evaluate changes in glucose levels in real time within living motor neurons from ALS models versus controls.
A challenging aspect is imaging the same set of neurons after changing the buffer. Leaving the sample on the microscope helps ensure that the VNC moves as little as possible.
Before beginning the dissections, turn on the imaging microscope and lasers. Then, collect a wandering third instar larva, rinse it with double distilled water, and place it in a drop of HL3 buffer on a previously prepared elastomer-lined dish. Next, under a dissecting microscope, use a pair of forceps to pin the anterior and posterior ends of the larva dorsal side up by carefully stretching the larva lengthwise with insect pins. Using a pair of angled iris scissors, make an incision just above the posterior pin. Then make a vertical cut from the incision towards the anterior end of the larva. After adding a few drops of HL3 buffer if needed, remove the trachea and the rest of the floating organs without disturbing the central nervous system. Then, pin the flaps, stretching the body wall, to expose the central nervous system while keeping the neuromuscular system intact. For image acquisition, use an upright confocal microscope with a 40 times water immersion lens. Then, select the 405 nanometer laser to excite the CFP. Next, optimize the acquisition parameters.